Hello everyone and welcome to today's webinar, Development of an Assay and Screening Platform to Study Molecular Glue Degraders. I am Cassie Soltman of LabRoots and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots and brought to you by Revity. You can learn more by visiting www.revity.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can use this Q&A box and we will assist you directly. I'd like to now welcome our speakers, Dr. Melanie Meister, Senior Scientist 2, AbbVie, Germany, and Dr. Johanna Huchting, Fraunhofer Institute for Translational Medicine and Pharmacology, ITMP, Discovery Research Screening Port. I'll now pass it over to you, Dr. Huchting, to begin the presentation.
Well, thank you so much for the kind introduction. The webinar today is focusing on the HGFR assay for the target RBM39 in the context of targeted protein degradation. And we will show you how we use the HGFR assay in our TPD projects. And for that reason, I will first give a brief introduction on targeted protein degradation and the assay technology. So in the field of targeted protein degradation, everything is about proximity-inducing drugs. And then some of those proximity-inducing drugs can actually be molecular degraders. But what are they actually? So a regular drug would block a function of, for example, an enzyme. But a proximity-inducing drug, what it does is it brings a target protein into close proximity of an effector protein. So in the native environment, those proteins would not interact with one another probably, or they would have low affinity for one another. But once the proximity-inducing drug is present, now those two proteins interact and they bind. And this can have a range of biological effects, and one of those effects being polyubiquitylation, when the effector protein is in fact an E3 ligase. So the target protein can be ubiquitylated, and that's then the signal for degradation via the proteasome of that target. And we work in this field in the context of a German cluster for future called proxy drugs, where many companies from the pharmaceutical industry or technology providers such as Revity are part of, and then academics are part of, and then us as Fraunhofer, we sit a little bit in between of academics and the industry, as we're a German organization that does applied research. And also AbbVie is part of this cluster, and Melanie Meister from AbbVie will also present later in this webinar. So we at Fraunhofer ITMP and the cluster of proxy drugs are interested in how can we actually discover and develop these proxy drugs. And at ITMP, we are developing a number of tools coming from different perspectives for molecular degrader discovery. And just to touch on a few, we develop informatic tools and models. We design chemical libraries. We have advanced models to test drugs and also proximity-inducing drugs and degraders. And we set up cell-based assays for screening and screen them, and that's what we're focusing on today. And part of that work then uses the HGFR technology, shown here as a no-wash sandwich immunoassay. So the basis of the HGFR assay is detection of proximity. So it's a thread-based detection. Once the thread donor and the thread acceptor are in close proximity, an energy pulse that is streamed into the system is donated from the donor fluorophore to the acceptor fluorophore, and I'm sure most of the audience will be well aware of that. And it uses a time-resolved measurement, which is important to reduce noise. And this can be used in a homogeneous setup, but how does this proximity now relate to identified drugs that degrade a target protein? So when we talk about targeted protein degradation, we actually want to measure the depletion of a protein inside a cell. And when coupling a donor and an acceptor fluorophore of a FRET pair to two antibodies that both detect different epitopes on the target protein, which can be RBM39, then those two fluorophores are in close proximity when they bind to the protein. So actually the signal that we read is directly related to the amount of protein that's present in the sample that we look at. So we can have a cell-based assay, we can lyse those cells after treatment, and we can directly now detect the protein level with this FRET pair labeled antibody pair. And if we have a molecular degrader present, we will see the depletion of the target protein directly reflected in the HGF ratio that we read. So that's the FRET ratio that we read then on our plate reader, and we see a dose response then of the molecular degraders that we analyze here. So we really can use this technology to analyze molecular degraders and determine the very basic parameters that we need for molecular degrader discovery and development, and that are the DC50 values, so the half maximal degradation concentration, and the % degradation that we can achieve with the highest dose. And with that short introduction, I will now hand over to Melanie from AbbVie and take back on then the screening part in the end.
Awesome. Thank you, Johanna, for your kind introduction. So welcome, everyone. What I would like to do today is to walk you through an application note that we have been writing with Gravity a couple of months ago, which is around the application of the RBM39 HTRS kit in our labs. And before we dive into the data, I will give you a short overview on RBM39, what it does, and why it might be of interest for the cancer field. And before we get started, let me briefly give a brief acknowledgement to the co-authors of this application note, which are Keith Ballard from Gravity, as well as my manager at AbbVie, Viktor Lakics. Okay, now, let's start. Okay, so let me tell you why we use RBM39 as a tool at AbbVie Neuroscience. RBM39 is much more of interest in the cancer research field. However, for us in neuroscience, it is a pretty cool tool to investigate the molecular glue-induced targeted degradation in neuronal cells. So RBM39 is also called RNA binding motif 39. It is a serine-arginine-rich RNA binding protein and is localized subcellularly to the nucleus, where it co-localizes with the core spliceosome subunits, which means that it is also functionally involved in the transcriptional coactivation and alternative RNA splicing. What makes it so interesting for the oncology research field is that RBM39 is upregulated in most cancers and is used in this field also as a prognostic biomarker for disease progression. The field has also shown that upon inhibition of RBM39, this is lethal to several cancers, such as lung, breast, or colorectal cancer, hence making it an excellent target for cancer therapy research. There is also Indisulam, which was recently in the literature, which is an aryl sulfonamide drug that has anti-cancer activity and which was shown to act as a molecular glue degrader. And what it does is it acts as a proximity-inducing drug. It binds to the colin-4 DDB1 decaf15 E3 ligase, connecting it to RBM39, which then leads ultimately to the ubiquitination of RBM39 and the proteasomal degradation. And this mechanism is so well described that it is really a nice tool for us to use this and transfer this to a neuronal setup. So, and here's what we did. So we took a neuroblastoma cell line, which is the SHSY5Y cells. We plate them to a 96 well at 50,000 cells per well, allow them to adhere for 24 hours, and then treat them with a concentration response in a half-log scheme, starting 10 micromolar, going down to 1 nanomolar. Let this incubate for 24 hours, and then we read either the HGF assay or a cell viability assay. We can also put these as a multiplex. And below this, you will also see, again, the HGF principle, how we measure RBM39 concentration or amounts within the cells. If it is there or if it's gone, we have a reduced amount of signal. And in this setup, we used, as I told you, Indisolam as a tool compound to monitor the RBM39 degradation. But we also saw this publication by Ruiz et al. From the Winter Lab, where they published DSM-1, which also is claimed to degrade RBM39, and they also published cyclin K degraders DSM-2, 3, and 4, which we used in our setup as a negative control. Now, if we use this assay workflow and treat our SHSY5 cells with Indisolam or the other DSM compounds in the concentration response, as I just introduced you to, we do see that if you have a look at the plot on the left, every color depicts a different drug. In the blue, you see always Indisolam in this slide. So Indisolam impacts the cell viability slightly, while the DSM molecules don't. So if we then have a look at the RBM39 degradation, we can clearly see that Indisolam induces the degradation of RBM39, despite the fact that we do have some impact on cell viability. We still reach DC50s of around 55 nanomolar with Indisolam. With DSM-1, this is a bit weaker or less efficient. We reach about 800 nanomolar DC50s. Both go down to around 20% RBM39 left, which makes it a degradation efficiency of around 80%. We do not see any degradation with the cyclin K specific molecular glue degraders DSM-2, 3, and 4. Now, we also wanted to have a closer look and compare the HGF performance to the regular Western blot, which is, of course, less well in throughput and much more labor intense. What we could see here, if you have a look at the Western blot, again, you see the dose response on top for Indisolam and DSM-1. You can see on the top RBM39. Please make sure to have a look at the top row of bands here. The lower one is unspecific. And then you also find vinculin, which we use as our housekeeping protein to normalize our data too. You can clearly see that both degrade RBM39. Even though we saw an impact on cell viability, the vinculin signal is still stable for the higher concentrations of Indisolam, and both compounds nicely are and reproducibly induce the dose-dependent degradation of RBM39. On the right, you will see the data plotted in relation to the vinculin housekeeping protein and normalized to DNSO. And you can nicely see the dose responses there as well. Now, if we have a really good and robust assay, which, by the way, gives us really solid set primes and solid performance from assay to assay, we thought we could also make use of this to understand the kinetics of the RBM39 degradation in SHSY5 cells a bit better. So we ran a time course, which started at 1 hour, 2 hours, and went on over 6, 18, 24, and 48 hours. And what we can clearly see is that we already reach a maximal degradation after around 6 hours. And also at longer time points, if you have a look at the brownish line, which represents 48 hours, there's no compensatory effect at longer time points, which means the cell does not counteract the degradation of RBM39 at that point in time, which clearly showed us that we can use HGRF assays, in this case for RBM39, to also understand the kinetic behavior of RBM39 degraders, which might be of interest further down the line during drug discovery and development. Final data slide is just to show you that this degradation of RBM39 really depends on the proteasome. So what we did here is we pre-treated cells for 1 hour with 1 micromolar epoxamycin, which is a proteasome activity inhibitor. And then after 1 hour, we treated with our regular indisulam dose response for 24 hours and read the RBM39 HGRF. And if you compare the DMSO-treated cells versus the ones that were pre-treated with epoxamycin, you can clearly see that epoxamycin blocks the RBM39 degradation and hence confirms that RBM39 degradation induced by indisulam depends on an active proteasome. Now coming to the last slide, and this is our summary. I hope I could convince you that using an HGRF as a fat-based technology really can quantify your cellular amounts of your biomolecular target, and that the RBM39 HGRF enabled us to monitor the RBM39 degradation in neuronal cells, in our case neuroblastoma cells. And overall, putting a focus on drug discovery, you really can use the HGRF assay to screen also through compound libraries for molecules that selectively target or degrade your target of interest. And for us at AbbVie, having such an assay is an essential part of a proper screening cascade. Finally, in-house, we will be and have been using the RBM39 HGRF, for example, to also further validate a nedylation deficient cell line, which is also part of the Proxy Drugs Consortium as a milestone. And with this, I will close and hand it back over to Johanna. I'm looking forward to the discussion. Thank you all.
Thank you, Melanie. So as you can already see, this is quite an integrative project where we all work hand in hand to really understand the technology, adapt it for our purposes, and really enhance our best use of the technology. And I will now continue showing how we now used the RBM39 HGRF in screening. As I might have mentioned in the introduction, we use the HGRF assay for a diverse set of targets in this proxy drugs project. And we have implemented a workflow that also integrates automation to really use this assay for screening. So the workflow that you see here uses 384 well plate format that in our shop, let's say, we usually use for screening. We have our libraries formatted in that way. And we integrate, for example, automated liquid handling, acoustic compound transfer, and use the devices that you see displayed here for that purpose. And as you can see here, this depicts a workflow where we do 20 hours of compound treatment, and that makes the assay a 3-day duration assay until from seeding until readout. But as you've seen in Melanie's presentation, where they did the time course study of the effect of indisulam degrading the RBM39 protein, it was actually quite quick. So actually the 6-hour dose response curve that we've seen showed already quite efficient degradation, and that's why in the case of RBM39 we were even able to short down the assay duration to a 2-day from seeding to readout, because we could do compound treatment for 6 hours only. So this one would then capture the more rapid degraders of the target that we're looking at, if you will. So first, when adapting the assay for our screening purposes, we looked at seeding density of the cells, and we were using the SHSY5Y cells as well, just as Melanie had also shown. So we titrate cells really at different seeding densities and complete the whole workflow that I had shown before, and then read the signal and try to understand where we sit in the linear range of the assay. We also have a cell line where we have genetically modified to express RBM39 from our construct, and we did the same with that cell line. And of course, we do read higher signals at lower cell densities here, just because the expression of RBM39 in that modified cell line obviously is higher than just the endogenous target in the SHSY5Y cells that you see in A. So really, we believe it's important to titrate the cell line that you plan to use in your assay and see for the linear range. As I said, we also use this assay with different targets. So then, of course, the antibodies change. So they need to be highly specific to your target, obviously. But the general workflow really is the same. And what I show you here is we also studied the androgen receptor in this cell line called MDA MB453. I've also depicted a little bit a picture from the microscope where you can see that seeding cells at 15K cells per well in a 384 well plate format obviously leads to quite a dense cell population there. But that's what it might take if you want to really understand and look at endogenous target and not a genetically modified version of your cell. We also look at IKZF1, a zinc finger transcription factor that is interesting to the targeted protein degradation field. And again, now D is showing genetically modified cells, so recombinant target expression, where you will find your linear range probably somewhere else than you will find with endogenous target. Moving on from there, we look at the assay window that we do achieve. So we look at a positive control degrader in the case of RBM39, as you have seen in Melanie's part of the webinar, that's endisolam. So we also treat our cells with endisolam at 6 hours. You can also do overnight treatment. That's not a problem. It just prolongs the total assay duration by a day. So you also see a concentration response on top, and then we look at the stability over the whole plate of the high and the low signal. We did the same in the example that I just talked about for the androgen receptor in the MDA MB453 cell line, where we have two different PROTACs at hand that are known to degrade the androgen receptor that we can use as positive controls. Only there we really do need overnight treatment, because those are not it's not a system where we see the rapid degradation that we see with endisolam in SHSY5Y cells. But both of those, in the end, with the same workflow, except for the difference in treatment duration, yielded about a threefold assay window in this screening setup, and quite nice Z prime values. If you're more from the field of biochemical screening assays, then you might aspire for higher Z prime values for the screening assay. But if you work in cell-based assays and try to really detect endogenous target, this is actually quite acceptable, I would say. So we were happy and moved on. So this is our screening layout now. So we really use the RBM39 HRF assay to do just a very regular library screening of single dose, full plates, 384 format. And as you can see, we have a library format that goes from columns 3 to 22 to include control degrader and vehicle columns. We also include in column 1 no cells, so we measure the background of the FRET signal that comes just from medium and DMSO and the antibodies. And then column 2 can be adjusted to really the target that you look at, whatever you need as an internal quality control or more positive negative controls that you have. This slide now shows the heat map that we obtained from our screen. So this is just an exemplary plate from the screening that I've described with the setup that I just mentioned. And what we read here is the FRET ratio. So it really shows the ratio of emission of the acceptor fluorophore over donor fluorophore. So really the detection of proximity of the two fluorophores that the FRET assay bases on is what is shown here. And as you can see in the columns 23 and 24 is our low and high control. So low 23 is endisolam. that should degrade RBM39 and should result in a low signal, and then 24 is just vehicle treated. And with that, we normalize all of our plate. As you can see, we obtain quite good signal inhibition, around 90%, so that's the degradation that indisulam then achieves. On this plate, we see around 2.5-fold assay window and a Z prime of 0.4 that we're happy with. And in total, out of 942 compounds that we had in this lower throughput screen, we identified 12 hits. And the hits were just very simply defined as showing above 50% signal inhibition at 6-hour treatment time point. So what I've marked here are the 2 hits that we find on this exemplary plate. And what we always do is we look at the donut channel for the whole plate. So this heat map now shows the values for the donut channel for the full plate. And you have seen, if I can go one slide back, you see this X in the first column where we really only measure the background of the technology. We could easily X this out because in the donut channel we see that there was no antibody dispense, so really just a dispensing error, really just a QC, and we take out that well. But other steps in QC that we do looking at the donut channel is that we really expect the donut channel to be quite similar across the full plate. And we do see a bit of deviation, and that I've marked here. So these 2 wells are deviating the most from the mean of the full plate. And we've looked back into our library what compounds we had in those wells, and those were in fact interfering compounds, and they absorb part of the emission fluorescence. So that's how we can really detect compounds that interfere with the technology. And we go back to our threat ratio heat map, where we initially had seen those 2 hits that were identified on this plate, and one of the hits that is marked now was actually then later on invalidated through a counter assay. And the other hit that we found was a well-known degrader of RBM39. So the library that we've screened here was actually a collection of compounds that we have curated from our internal libraries, but also from purchased known degraders to really calibrate and understand the assays that we integrate into our proxy drugs toolbox. And so this library is spiked with degraders of targets that we are going to look at, and so it is also spiked with degraders of RBM39. So we can really analyze the performance of the assay. And we do see here that we capture with this screening the compounds that we are supposed to capture. This known degrader of RBM39 survived all of our counter and QC steps, and we were able to invalidate other wells or compounds through our QC and also our counter assay. So what I hope we could show you in this webinar was that we do need cell-based assays to screen for molecular degraders, because molecular degraders need the whole cellular machinery to really effect, in the end, protein degradation via the proteasome, and that the HGF assay can detect endogenous target, it can quantify endogenous target in a way that is adaptable for screening, and we have developed a workflow that is, in the end, applicable to diverse targets. With that, I would like to thank all of the collaborators on the RBM39 story that we did within Proxy Drugs, my colleagues here at ITMP, and Melanie for the nice collaboration that we have in this consortium. Thank you all for listening. I'm happy to take any questions for the discussion.
Thank you, Dr. Meister and Dr. Huchting, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started here. Our first question is for you, Melanie. It says, how much time does the HTRF take in comparison to a Western blot?
Thank you for the question. So the HTRF has much less hands-on time than a Western blot. You don't need to do protein amount estimation and so on. So what takes most of the time is actually the lysis of the cells and then the transfer to the 384 well, and then adding the antibodies. That's basically the 3 steps that you will do. And then you incubate this overnight. So depending on plate numbers and whether you have an automation that will do your plate handling, for me, usually I have 1 to 4 plates, and I do not use an automation for 1 to 4 plates. So the pipetting part is like around 90 minutes, and then the day after, it's basically just putting it in the plate reader and waiting for 2 minutes for the result.
Okay, excellent. Thank you. Our next question we'll ask over to you, Johanna. It's asking which compounds are interfering in your HTRF screening?
So as I've shown, we detect interfering compounds in the donor channel, for example. So those are compounds that absorb fluorescence that we want to read. And so it's chromophore compounds, that colored compounds that would just interfere with any fluorescence-based readout type assay, I would say. So those I would flag as interfering compounds here. Yeah.
Wonderful, thank you. All right, back over to you, Melanie. This question says, you shared information around the DC50 of indisulam after 24 hours. What about DCmax and Dmax?
So the DC50 of indisulam in the SHS-554 cells is in our hands around 55 nanomolar. The Dmax we observe is around 90%, and the DCmax is at 10 micromolar. But in this case, please keep in mind we are not controlling for cell viability in this case. And if you would want to control for cell viability, we would want to either make this to the protein amount, or we could run a housekeeping HTRF such as tubulin, for example, in comparison. And also keep in mind that the DC50 of indisulam is in a sub-micromolar range, while the toxicity that is beyond 25% of impacting the cell viability only comes in the single-digit or double-digit micromolar range. So 2 separate things that we can observe.
Wonderful, thank you. All right, our next question here says, this is for you, Johanna. This says, which are typical false positives that you find in your HTRF screening?
So I would say this depends really on the compound incubation time that you use. So when you can work with short compound incubation times, as I would say we did in this RBM39 case with 6 hours, that I would say is still on the short side. You definitely limit your false positive rate by that. So if we look then at like a 24-hour overnight compound incubation, we do find more false positives. And those are then compounds that really just inhibit protein synthesis. So your typical protein synthesis inhibitors you would find PI3K mTOR inhibitor, you would find topoisomerase inhibitors. So compounds that would then also very early show up with cytotoxic signals, I would say.
Wonderful, thank you. All right, back over to you, Melanie. You shared that you run cell viability and HTRF on 2 separate plates. Would you be able to multiplex them as well? And are there alternative readouts for tracking cell viability in this setup?
Thank you for this question. So I tried to multiplex the cell titer blue and the HTRF. So the cell titer blue works in live cells. So you incubate the cell titer blue on the cells, and after 3 hours you will read it, and then you would wash the cells with PBS and then immediately process them for the HTRF. So that works. However, what we also could do is if you have cells that grow like SHS-by5Y cells and you are not limited by cell number, what I usually refer to is running duplicate plates, because I did not want the assays to cross-interfere with each other when it came to the readout in the end. What you always can think of is running an HGFR housekeeping gene, such as tubulin. This is something we usually do for our iPSC-derived neurons. That should give you a readout on cell viability as well.
Sorry, I was muted. Excellent, thank you. And then our next question, back over to you, Johanna. This says, in your workflow, you have shown a medium aspiration step before cell lysis. Is this strictly necessary?
So it's not strictly necessary. I would say you could add lysis buffer to the treatment medium and not remove any of that. We typically don't do that because in general, we want to get rid of the compound that's in the treatment medium, because that might then have stronger interference with the readout technology, so the HGRF then in the end. So it's possible to do that, definitely, but if it's possible to just take off your treatment medium, I would always advise to do that, and actually not just in this setup, but in all kinds of different setups, to really get rid of the compound that's in the medium still.
Wonderful, thank you. All right, our next question here says, how do I take into account the cell death that is caused by the degrader?
So we briefly touched this, right? You can multiplex with cell viability assay, which will give you an answer on how toxic your compound would be. You can also run a housekeeping gene or protein as an HTRF, but usually you should keep in mind that when you run these RBM39 HGRFs or any target HGRF and compare to cell viability, these are two different things, right? So you will get your DC50s for your degradation, and you will need to cross-check how far away is my EC50 for the cell toxicity. Johanna, I don't know if you want to add to this from your perspective.
Yeah, I would probably again mention the time that we look at, so the compound treatment time. So usually cytotoxic compounds, I mean, there's few compounds that are super quick in their ability to kill cells or cell growth. With the treatment time that we look at, we don't look at cell growth, right? So compounds that just inhibit cell growth are not false positives here. Or taking away of the signal of the degrader or interfering with that, however you want to put that. So if you really have short treatment times, I wouldn't worry about that too much. But just as Melanie said, also, of course, you do have to put a counter against that.
Excellent, thank you both. All right, our next question here says, RBM39 expression is unregulated in hypoxic tumors. What is the kinetics of induzilam to RBM39 in hypoxic conditions?
So from our side, I can say that this is not something that we have studied. So I definitely can't answer that question. So we did not look into the specific parameters of induzilam, and we did not study any specific models. Let's say we were more focusing on how to really introduce the technology into the screening environment and validate the assay in our screening setups. I don't know if Melanie has any insights into that, but we didn't specifically look into that.
No, I like the question, and I like the tendency towards the cancer field it has, but I also, working in neuroscience, we work usually in neurons or in a neuron type cell line such as SHS-by5Y, so we usually do not look at hypoxic conditions. I'm sorry, I cannot answer that question.
Excellent, thank you. Not a problem at all. All right, so it looks like we have time to fit in one more question here. This last question we'll ask today says, which are the most common optimization steps to be performed to have a robust HTRF assay?
So I can give it a go and start, and Melanie, you can add if you like.
Yeah.
So what we see, as I've shown also in the presentation of this webinar, you really need to make sure that you're within the linear range with your cell numbers. So seeding cell titration is important because it really differs from target to target, from model to model. You have to be sure where you're at in that linear range. And then you need to have I would say, a potent positive control to be able to calibrate your parameters and be sure of what you're seeing. And then I would say the liquid handling steps are actually quite critical, because you're working with small amounts and you're working with endogenous targets. So wherever you have a slight variation, just because of liquid handling variation, it would clearly show in your signals. You would really see variation in your signals. So we did spend time developing our workflow and really being sure of the liquid handling steps. So that's something that you really need. Apart from that, I would say the signal's quite stable. So you can do, I'm not sure if we tested with this RBM39, but usually in the HRF assay, you can do a readout after 2 hours of incubation with the antibody and the cell lysate, or overnight. It's a stable signal. You can do both. So that are not really steps that you would have to optimize, but you have to, of course, have a homogeneous cell lysate that you transfer to your detection plate. We feel that it's good to work with a detection plate and to really have a transfer of the cell lysate. But of course, again, when you transfer cell lysate, you need to be sure of your liquid handling steps.
Yeah, I agree to everything you said, Johanna. Thank you for that. And just something out of the lab that we usually do if we have the liquid handling established, one cross-check before we go live with the screening is something we do is a plate uniformity, just high, medium, low controls all over the plates in different variations. And then you will see how well your liquid handling is also working. So that's one thing we also usually do. And also, just for robustness, we usually calculate all the parameters such as Z prime, CV% that are out there.
Excellent. Thank you again for both of your time today and your important research. We would also like to thank LabRoots and our sponsor, Revvity, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers or Revvity via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.